Thank you very much, and thank you. It's lovely to see people again. I only wish those online if we could see you too, because it would be nice to see who's there. Apologies for my voice. After all this time of COVID, I've actually got a cold, so if I'm a bit hoarse, I apologise for that. So I've been asked to talk about the guidelines, international guidelines, to look at how similar and how different they are. And I'm going to just start with my conflict statements. Now, the first one on here is probably the most relevant for writing treatment guidelines, but actually the second one on the list is perhaps more relevant for my talk today. So, as Genevieve just said, I am the Vice Chair of BEVA, but I've also been a member of its antiretroviral treatment writing guidelines writing group for many years. And over that time, we have spent a lot of time trying to make our guidelines as robust as possible. So I hope you'll just forgive me if I'm a little bit partisan towards these. So as background, when antiretroviral treatments were first introduced in the early 1990s, there really weren't many drugs around, and therefore there wasn't a huge amount of need for treatment guidelines. And those that did exist tended to focus on the bigger, more strategic questions, such as when to start. And of course, you know, that was in an era where treatments were perhaps not so effective as they are now, and where actually they weren't so tolerable as well. Now, obviously, as the number of drugs increased and the number of classes also increased, then the potential for people to be on treatments that were suboptimal and, you know, with not particularly much evidence to support them really increased. Sorry. And at the same time, you know, with little guidance on their use, you could see that the potential for treatment costs to spiral out of control also increased tremendously. So treatment guidelines really have been hugely influential in ensuring that everybody with HIV has access to at least a very high quality of care. And certainly for those of us in the UK, where treatment is you know, it's free at the choice at the point at which the person receives it, but it is funded by the National Health Service. So the other benefit of guidelines for us is that they've tried to control and make sure that the treatments that people receive are not only effective, but they're cost effective. So what I want to talk about today is two things. I'm going to start by looking at the 4 of the major treatment guidelines and looking at what they say. And I hope that you will appreciate, given that some of these stretch to hundreds of pages long, I'm just going to focus on the questions about when to start and what to start. And both of these will be in the context of relatively uncomplicated HIV presentation. And then I'm going to go back and look at the guidelines and have a think about how the guidelines were generated. And this is where perhaps I'm going to bring my more methodological perspective to the table. So I'm not a clinician, and you know, I'm surrounded by many eminent clinicians today who know far more about it than me. But I'll look at the methodologies that the panel use and maybe think about whether some of the differences there can then lead to differences in the guidelines. So as I said, I'm going to talk about 4 of the guidelines. I'm going to focus on guidelines that really target people in resource-rich settings. So for that reason, I'm not going to look at the excellent WHO guidelines. But if I look back in the oldest first, I'm going to start by reviewing the IAS USA panel guidelines. They were published in October 2020. Then the recent update to the EACS guidelines published a year later. Then the USDHHS guidelines, which were updated at the start of this year. And then finally the BEVA guidelines. Now, our most recent published ones are actually quite out of date now, but what we are doing is updating ours, and they're currently out for public consultation. So the guidelines I'll report to you today just have that caveat attached to them that they may be tweaked in a little way before they're finalised next month. So let me start with the big question, when to start. And I think if we focus on the bigger picture, if you look at the text here about what all those guidelines say about when to start, you can see that pretty much all of the guidelines are in agreement. Everybody with HIV should be offered antiretroviral therapy regardless of what their CD4 count is. And there's no real difference between them. But I think the devil is in the detail here, and where you start to see the differences applying is when you read into the text about how urgently the recommendation is to start treatment. So if we look at the IAS guidelines, they say that initiation of ART is recommended as soon as possible after diagnosis, including immediately after diagnosis if the patient is ready to commit to treatment. For EACTS recognise that there is now an accumulation of evidence to support the fact that immediate same-day antiretroviral treatment is feasible and it's acceptable to people with HIV. But importantly, I think, for me, what EACTS says is that actually before you start treatment, you really should be assessing the readiness of that person to start treatment. And that means that it's ensuring that people with HIV can express their own preferences and don't feel pressurized to start treatment unless there is a clinical need. And if you're not familiar with the EACTS guidelines, there is a really good tool in there in terms of how to assess readiness. Now, the DHHS guidelines recommends initiating ART immediately or as soon as possible after diagnosis, and BEVA pretty much follows EACTS in terms that readiness to start is the important thing to assess first. And in fact, BEVA now points readers to the EACTS tool for assessing that. Now, I've underlined at the bottom there the fact that the BEVA guidelines also make a point that there is a lack of proven benefit for same-day ART in a UK setting, and I'm going to come back to that later on. So in terms of when to start then, I think it's pretty obvious to me that all of those guidelines pretty much are in broad agreement. So everybody with HIV should be offered treatment regardless of the CD4 counts. The difference really is in terms of how soon that offer is made. But what's very reassuring, and I think is nice to see over the last few years, is that actually the person's own preferences have really come to the fore in some of these guidelines now. And readiness to start is emphasised, that people should assess that first before starting treatment. Okay, what to start? Well, I've split this up into the regimens that include INSTIs and then those that include PIs and non-nukes. And obviously, there's issues around co-formulation, generic drugs, that will be very much geographically defined. So I'm not really going to focus on that. I'm just going to talk about the drugs themselves this morning. So if we look at the INSTIs, there are two regimens on there which are broadly recommended by all four of the panels, and that's bictegravir, TAF, FDC, and dolutegravir with TDF, TAF, and either 3TC or FTC. They're all recommended without any caveats. And then we've got three regimes, the two dolutegravir, the dual dolutegravir 3TC one, and then dolutegravir with the bacavir 3TC, and raltegravir with TDF, TAF, and FTC. All of those are broadly recommended, but with caveats, so in certain clinical circumstances. And as you might imagine, those are pretty much the same across the panels. The only one that sticks out as being slightly odd is elvitegravir, and that's one that DHHS recommends, but the other three panels don't. So for the INSTIs, pretty much broad agreement now, I would say. For the PIs, which is in green on this, which I think you can just see, there's only one regime that is recommended across all the panels, and that's the boosted darunavir with TDF, TAF, and XDC. And all of those, again, are recommended, but in certain clinical cases where perhaps the first recommended option isn't feasible. And then the other four PI regimes on this list are not recommended by any of the panels apart from DHHS, albeit, again, in certain clinical cases and not as a routine sort of first-line choice. If you look at the non-nukes at the bottom, then the first two on there, so there is broad recommendation for use of doravirine and for efavirenz, both with TDF, TAF, and XTC. Again, for most of those panels, there are caveats for when they should and shouldn't be used. But the only one that sticks out really as being a little bit odd from the non-nukes is rilpivirine, and the one group that doesn't recommend that is BEVA, but the other three panels do. So overall, I would say that increasingly, and I think, if I'd done this talk maybe 5 years ago, this wouldn't have been the case, but increasingly the guidelines seem to be in fairly broad agreement about the recommendations. For the INSTIs, certainly, the only one sticking out is elvitegravir. For the PIs, really, it's only the boosted darunavir regimen that is the one that's universally recommended, with DHHS suggesting some other ones. And for the non-nukes, the one that sticks out really is rilpivirine, but there is possible use of doravirine and efavirenz. So I think, broadly, these guidelines are coming together. So why might the guidelines differ? What is it about those panels that comes up with different recommendations? Well, I think that traditionally, and this me being a little bit cynical, we perhaps tended to think of guideline panels as a group of people who sat around a table, maybe with their cups of tea or their cups of coffee or maybe even a glass of wine or so, and they reviewed the data they'd seen and came up with these recommendations. And of course, to be fair, that's probably not far from the truth, particularly in the early days, because many of the traditional guidelines were very opaque, so it was hard to know, how people had done their literature review, what terms had they used, how had they brought together the differing pieces of evidence, how had they balanced up and weighed evidence from, say, a randomized trial with a case control study, or different outcomes of treatment, so virological failure with mortality, say. And of course, because of that, what it meant was that given exactly the same evidence, you could have different guideline panels coming up with very, very different recommendations. But I think what we're starting to see is that the panels now really recognize the value of transparency. And of course, because of that, everybody's methods are published, and you can see that the guidelines, I think, are coming much more closely together. And of course, we're not in a vacuum. Everybody can read the other guidelines, so we know what the general direction of flow for these guidelines is. So let's have a quick look at the methodology, and apologies for anybody online or in the room who's a member of one of these panels if I've misunderstood anything. I've taken this all from sort of online publications or the actual papers themselves. So the IAS, it was one of the earliest ones. It was established in 1995. Its members are selected by the Board of Directors. I looked to see whether there was a statement of inclusion of people with HIV, and to me there wasn't anything there, and I scanned through the names, and I may have missed people, but I didn't see anybody with HIV on that panel. Conflicts, I think, of all of the panels, this is the one that has perhaps the most stringent and rigid guidelines in that participants don't participate in industry promotional activities during their tenure. They can still participate in non-promotional activities, but that is at least reported, and you can see people's conflicts. And the aim of these is that adults in settings in which most antiretroviral drugs are either available or they're in late-stage development. And so I guess that's Europe, it's North America, it's Australia, New Zealand, etc. Now, for the panel, they consider new published evidence, but they also include presentations at peer-reviewed conferences and safety reports, so pretty much a broad range of evidence. The literature searches are reviewed by 2 authors, and then the search strategies are now published online. I think that's new for the last few years. And the recommendation is by full panel consensus. And let's just look at these, because you'll see this is a fairly standard sort of classification. But at the top of that table there, you have the rating for the strength of the recommendation, which is either classified as A, B, or C. So A is a strong support, B is a moderate support, and C is a limited support. And then the quality of the evidence is categorized as 1, 2, or 3. So category 1 would be the evidence comes from at least one randomized trial. Category 2 is where there's no trial data, but it's all from cohorts or case control studies. And then category 3 is expert opinion. And the categories 1 and 2 are subdivided as well according to whether the evidence has been published in journals or whether it's just available in abstract form. Now, EACS, I think EACS is actually the most recent of the panels. Their first guidelines were published in 2005. The panel is overseen by a guidelines chair from the governing board of EACS, and the panel membership is rotated following standard operating procedures. I actually couldn't find out what those standard operating procedures were, but I probably ought to ask some of the EACS members in the room. The inclusion of people with HIV on the panels, there wasn't any clear statement about that online, but looking through the panels themselves, you can see that at least one community member is included on all of them. And potential conflicts, declarations of conflict are available on request, so you can request that information. Now, the aim and the context for these guidelines is obviously Europe. So Europe includes a lot of different countries with lots of different healthcare systems. So these guidelines, by definition, are very broad because they're aiming to follow everybody. The recommendations are evidence-based where possible, but they're based on expert opinion otherwise. There aren't any formal grades of evidence, and the panel decision is a consensus one. But interestingly, they don't publish the results of any voting or any discrepancies, so we don't really know what the strength of support was for each of the guidelines. And it wasn't clear to me whether the guidelines are subject to public consultation or peer review before they're published. For DHHS, there's 50 voting members on the panel, and interestingly and unusually compared to the others, this also includes representatives from a range of regulators, so you've got a slightly different perspective coming in here. The panel does include 4 to 5 community representatives, and the annual financial disclosures are published online, so you can see all of that information. But these guidelines are really firmly aimed, I think, at US healthcare providers, so within that healthcare setting there. So this is not too different from IAS. They consider peer-reviewed published studies or data from FDA drug labels. Where they start to differ is in terms of whether they consider abstracts. And it seems that really it's only if it relates to patient safety, then they will also consider unpublished data from conferences or safety warnings. There is a 2-week comment period, and the guidelines, the recommendations are rated in the same way, very similar way to the IAS ones. So with the ABC for the strength of recommendation, and then the class 1, 2 or 3 for the quality of the evidence. So finally, BEVA. Well, BEVA, our guidelines were first published in 1997. They are accredited by the National Institute for Health and Clinical Excellence, which is NICE, which is in the UK. Now, that may not mean much to many people in the room or online, but certainly for those of us in the UK, this introduces quite a lot of complexities because we have to meet their standards for guidelines. But what it means is when the treatment guidelines are accepted and approved, then it may become easier for people to prescribe some of the drugs, particularly if they're not the cheapest options out there. So for our panels, the subcommittee and all writing groups do include community representatives. We have our potential conflict policy is dictated really by NICE, so the writing group chair has to be free of any connection with pharma in the 12 months prior to taking up the role. But all of the other members, we report financial disclosures annually. And again, they are published, so they are available to look at. And the aim of these is definitely at UK NHS healthcare providers primarily. So if you want to know more about the BEVA approach, it's published on the website. There is a rather short and sweet 70-page guideline development manual or something if you get bored and want to read it. But I want to summarise the methodology because we do now start to differ a little bit from some of the other panels in that we will commission a systematic review to address a PICO question, and we then link the results from that systematic review to our recommendations using the grade system, and I'll explain what I mean by that. The guidelines are person-centred, so they use appropriate language and take account of equality, financial implications and patient choice. And as said, they're open to public review, and they are also sent out for peer review. So what is grade? Because you may not be aware of this. Well, grade, I mean, if you're involved with systematic reviews, you may be familiar with grade, but it is a process for developing and presenting evidence from systematic reviews and treatment guidelines. And what grade requires us to do is to identify the question first and then do a systematic review of the literature relating to that question, and then the results from that determine the strength of the evidence for that recommendation. Now, that may not sound that different to other panels, but in fact, because we are now forced to do this at the start, and we use the PICO approach for identifying the question, it means we have to be very clear at the outset what our patient or population group is, what our intervention is, what the comparator is, and what the outcome is. And it's probably the outcome of that which is perhaps the most interesting, because with grade, you're supposed to pick outcomes that are of importance to patients primarily. And so things like mortality, morbidity, adverse events, say, may well rate higher than a change in CD4 count, even though clinicians think the CD4 count may be more important, because it's about who it's important to. And so we have to define that before we actually do the guidelines. And just as an example, for this round of guidelines, we asked the panel in advance to rank 15 different outcomes as to how important they felt they were. And these are the 10, so 5 critical outcomes and 5 important ones. And these actually have changed since we did this 5 years ago, 6 years ago. So some of the endpoints around virological failure and adverse events and resistance, they've moved up the list, whereas other things have moved down. And that's because over time, what we see as important from a first-line regime has changed. And so, you can already see that even with the same set of evidence, our guidelines could change because our views about what's important has changed. Now, GRADE then takes the results, and again, it assesses the quality of evidence in a similar way to the other studies. Now we've got 4 categories, so high, moderate, low quality, and then expert opinion. But where GRADE differs is it recognises that actually good quality data from a well-controlled, a well-designed observational study may actually give you better information than a poorly designed randomized trial. So now what you see is that those top ratings will allow you to include data evidence from RCTs as well as observational studies. But GRADE tells you how to do that. So if we start with a randomized trial, say, we would initially say that's high quality evidence, but we can downgrade that if we think there's a risk of bias, if there's results from different trials are inconsistent, if it's not addressing the PICO question, so if it's indirect, we could downgrade it, if the confidence interval is wide, or if there's a risk of publication bias. But equally, if we start with an observational study, they will start as low evidence, but we can also upgrade those. So if the effect is strong, if there's a dose-response relationship, or if we can see that all of the confounders have either been addressed, or even if we could address them, they would actually strengthen the association, then we can upgrade the evidence. And so you can see there that you have a bit of sort of upping and downing with the quality of evidence. And what we then do with our strength of recommendation is we go to a binary categorization. So a grade 1 recommendation would be a strong recommendation. That's where the benefits clearly outweigh the risks. And in that case, we use the language "we recommend." Or we can have a grade 2 recommendation, which is a weaker one where the risks and benefits are more balanced. And there we use the language "we suggest." So we make sure that the language is compatible. And then we have a final point, which is a good practice point, which is it's not based on any evidence per se, but it's where the action is so obvious that nobody would question it. So, for example, our recommendation that people with HIV should be involved in decisions about their own care is a good practice point. Everybody agrees it's valuable. We don't need evidence to support that. So I've explained the methodologies. Can they explain some of the differences? Well, if we think to when to start, and particularly the same-day ARC question, why is it that BEVA won't actually make a recommendation for that? Well, when you look at the evidence that's there is some trials, there's some observational studies, but actually none of them relate to our population, none of them relate to exactly that question if we were to write it in a PICO format. And so for us, when we reviewed the evidence, it was classed as low quality because of the risk of bias, inconsistency, indirectness. And that's why in our guidelines we make a note that people should recognise the lack of proven benefit for it in the UK situation. Now, of course, if a person wants treatment, that's also then recommended. So again, it brings the patient decision in. And in terms of what to start, well, actually, there weren't that many differences anymore, but I think what you can probably already see is that the difference is probably between the DHHS guidelines and the others, probably reflects the needs of the different healthcare systems. So it's really something probably as simple as that. But certainly where perhaps BEVA differs is around the priorities for outcomes and the fact that we've rated different outcomes higher than other guideline committees have. So to summarise then, I think what we can see is that the guidelines have become more homogeneous over time. There's very few differences now between them. Where the differences do exist, I suspect they're largely driven by the context for the guidelines, but also the methodology there. But what's really refreshing now is the guidelines are all starting to demonstrate far more increased transparency about the decision-making process. And also for me, I was really pleased to see now the much more obvious involvement of people with HIV into the guideline development process, but also the recognition that involving people with HIV into decisions about their own health is so important. So that's really my very whistle-stop tour. I want to thank everybody, particularly Laura, who gave me a few slides and checked over to make sure I'd interpreted the guidelines correctly. I want to also thank the Guidelines Subcommittee, particularly Claire Van Halsema, and the ARC Guidelines Writing Group. I mean, this group does a huge amount of work over the years to try to get our systems robust and in place. So thank you very much.